This is the cytoskeleton Introduction 2, focusing on intermediate filaments. Here we're looking at about 10 cells, each of which are fluorescently labeled for 2 different proteins. In blue, we have the cell boundary. And in green, we've got keratin, which we'll hear a lot about very soon. So that's in green, and this is one of the main components of intermediate filaments in many cells. One of the main things that you'll see is that the intermediate filaments of keratin extend throughout the cytoplasm. So it's a big theme. This would be the nucleus here, which does not have intermediate filaments in a large portion of it. And we see the edge of the cells outlined here with this blue fluorescent stain. So basically the intermediate filaments will surround the nuclei and extend through the cytoplasm From our last video, you'll recall that there are particular connection sites of the intermediate filaments between cells, and those are called desmosomes. And if you look at the figure over here just for a second, see if you can find any of those locations for the desmosomes. Remember, these are cell-cell junctions. And this is a mechanical link between cells. So here's a couple of the locations for the desmosomes. Right along here, you can see the intermediate filaments basically extending between the cells, kind of creepy looking. Lots of other locations here. And basically, if you look at pretty much every single one of these cells, you'll see that they have several desmosomes. Oh, that's not what we were looking for.
Eukaryotic cells contain a complex network of filaments, intermediate filaments, microtubules, and actin filaments, that provide the cells with strength, structure, and movement. Although all eukaryotic cells contain microtubules and actin filaments, intermediate filaments are found only in vertebrates and a number of other soft-bodied animals. Intermediate filaments are found in animal cells that require a lot of strength, such as the epithelial cells of the skin. Some of these filaments span the length of the cell, connecting cell-cell junctions called desmosomes. These cables of intermediate filaments have a high tensile strength. Without these filaments, stretching or pressure on the epithelial sheet would cause it to rupture. Each filament is rope-like, consisting of 8 thinner strands made of a precise hierarchical arrangement of protein subunits. At the lowest level, 2 monomers associate with each other to create a twisted dimer. 2 dimers then line up to form a staggered tetramer. Note that the 2 dimers are arranged in opposite orientations with their amino terminal ends away from each other, so that the 2 ends of the tetramer are indistinguishable. Tetramers then link end to end, thus building up one strand of an intermediate filament. A total of 8 strands stack together and twist around each other to create the intermediate filament. This stacking provides the extensive lateral contacts between the strands that give the filament its remarkable mechanical strength. An electron micrograph shows the appearance of intermediate filaments that have been assembled in a test tube.
So here we're going to talk about some of the details about intermediate filament structure. Remember that this is a rope-like structure, like a big cable. It's very strong. and there are many strands that are twisted together and provide this mechanical strength and tensile strength. The strands are made up of many intermediate filament proteins. They're put together in a very specific order, including the central rod domain, which is approximately here, for a single protein subunit of the intermediate filaments. The central rod domain is very similar across all of the intermediate filaments within a particular category, a particular type of intermediate filament. On the other hand, the terminal ends, indicated by the arrows, those vary quite a bit amongst the different intermediate filaments. This can determine what the intermediate filaments will interact with and bind to as far as other cellular components or locations. And basically, we see that there's this alpha helical structure of this monomer of an intermediate filament. It has an amino terminal and a carboxyl terminal highlighted here. And these assemble into coiled-coiled dimers, which run in the same direction, where the two amino termini are in one location, and then the carboxyl termini are in this disparate location. And these twist together to make this dimer protein. Then there's a staggered tetramer of several coiled dimers put together. These are overlapping, as you can see, and these run in opposite directions. These are the soluble parts of the intermediate filaments, or the dimers and the tetramers. And basically these can be put together to make a lateral association of 8 tetramers. And finally, 8 tetramers are added to each other, and this makes a growing filament. In other words, the final rope-like intermediate filament here on the bottom. A few other things to notice about the intermediate filament is that this structure is not polar. In other words, there's not one side is not distinct from the other, and this is due to the fact of how we have the opposite-oriented tetramers. And basically, this is distinct from other cytoskeleton components, which we will talk about later, such as the microtubules and actin. It's very important for them that they're polar as far as their functionality. However, intermediate filaments are not polar. And one additional thing to point out is that all these interactions between the different units, the different subunits, monomers, dimers, etc., all of those are due to non-covalent binding between proteins, which is what we would normally expect for protein-protein interactions, that they are non-covalent. A couple other things I'd like to point out about the protein structure is when we talked about the terminal ends here, these are unstructured regions, whereas the central rod domain, again, is an alpha helix, and these will all be similar size to one another, as well as similar composition of amino acids. Intermediate filaments are found in a variety of cells which are subjected to mechanical stress, and I want to talk about those briefly. So mechanical stress is high in the axons of nerve cells. Those thin extensions are fragile, and so they have a lot of mechanical stress. In these cells, the intermediate filaments are called neurofilaments. Each of these types of intermediate filaments, which I talk about, that are cell-specific, have different protein subunits. Another sort of cell that is subject to mechanical stress is muscle cells. And in these cells, they're called viamentin filaments. Finally, in epithelial cells, there are a variety of different types of keratin filaments. All three of these sorts of intermediate filaments are found in the cytoplasm. And you may have heard of keratin before in hair and skin cells. That's one type of keratin which is found. All epithelial cells in vertebrate organisms have specialized versions of keratin. There's different versions in the tongue or the cornea and other epithelial cells as well. Finally, all eukaryotic cells have a nucleus, and remember that there is the nuclear lamina, which is made of intermediate filaments. So this is another location we'll find intermediate filaments, is in the nuclear envelope. So inside the nucleus, and again, this is specifically in the nuclear lamina. The picture that is shown here, which we haven't talked about just yet, is looking at a cross-section of skin. On the left, we see a cross-section of wild-type or normal skin layers. This is a microscopic image. And on the right, we see a mutation in a type of keratin in a mouse. And you can see that there's some major disruptions. This leads to substantial blistering and is a very serious condition. There are also some human mutations in intermediate filaments which can cause some major problems and have a substantial decrease in life expectancy. The last example I want to talk about is another human condition which has to do with mutations in intermediate filaments. This is a condition called progeria, and this is an extremely rapid aging disease, as you can see. And the individual shown in the figure here, this is a child, maybe just 5 or 6 years old, and already has some incredibly advanced signs of aging, and probably has a life expectancy of about 8 years. This is due to mutations in certain intermediate filaments, which make up the nuclear lamina. And it's not known exactly how this leads to such rapid aging, but it is thought that the DNA is less stable. There's genomic instability due to the lack of the intermediate filaments. Chromosomes are broken very easily, and again, this can lead to a variety of different effects. If we zoom in to check out what the nuclear lamina look like in the intermediate filaments, look down at the lower part here. So here we have the cell, the nucleus, and basically if we look into the nuclear envelope here, then notice in blue, that's where we see the nuclear lamina. Those are the intermediate filaments. And then we see there's a very close association with chromosomes and chromatin. So you can imagine if the nuclear lamina was not properly protecting the nucleus and the chromosomes from mechanical stress, the chromosomes would be very fragile and could be damaged. And that wraps up this video on the introduction to the cytoskeleton 2.